Good afternoon, and welcome to this season two, probably the last episode of the season of this Chai Time series. And it's a lovely, sleety, snowy day that we've had in Cambridge. I've had my colleagues from India calling me up saying, "Oh, it's so cute that it snows there." I'm like, "No, it's dark, cold, miserable," and the snow has kind of turned to sleet now. So we're in this kind of horrible winter season now, but we're all positive, we're all jovial. Christmas is coming up, the vaccine is here, and lots of reasons to be cheerful. We're out of lockdown, just about semi-lockdown, and we're looking forward to getting out and about again. And on that note, I really wanted to introduce the chair of our current Chai Time. He has been a previous chair with O2H, and a good friend of mine and our groups over many years is Andy Richards. He's been single-handedly, since I was a young whippersnapper, leading the UK biotech industry, and it's very great of him to chair this session and to introduce what I believe is the next generation of fascinating biotechs that are emerging from our UK ecosystem. So without further ado, I'd like to hand over to Andy Richards.
Hey, Sunil, thanks. And yeah, thank you. That's very, very kind of you. I've never thought of you as a whippersnapper. So welcome everyone. I can see names coming up. Great set of attendees, many of whom I know, some I haven't seen for ages. Barbara, lovely to see your name there. And just hope that everyone feels that able to ask questions. This is about your questions, so please put them on the Q&A. I will try to, on the chat, I will try to pick those up and ask questions and get them asked. But I think this is a really good and interesting session. We've got three really interesting, diverse, exciting therapeutics drug discovery companies today. We've got one with a very clear therapeutic focus in the dermatology area. We've got one based on a next generation antibody drug conjugate platform targeting towards cancer cells, and a third which is an exciting AI platform based on quantum physics, with a platform that could be applied to many, many, many different targets. And hence has that challenge of where does one go with that. So great to introduce Tim from Inforderm, Miriam from Spirea, and Vid from Kuanu. I am going to let them all speak for 5 to 7 minutes, giving you a bit of a what problem are they trying to solve and how they're trying to solve it and where have they got in that journey. And then I'll start throwing open to questions. Please do ask questions on the way, and let's get going. So Tim, over to you to start with, if that's okay.
Great, thanks Andy, and hello everyone. I'm talking to you from the not snowy wastes of North London, unlike Cambridge, which seems to have had a little bit of snow. So I'm here to tell you a little bit about Inforderm, and as Andy said, we're a new company. Focused very much on the dermatology space, but with the challenge to remain focused on dermatology. I'll explain why in a moment, but we spun out of the University of Dundee just back in the late summer, and we've just recently closed a seed round, and I'll come on to that. But why is Inforderm in existence? What, as Andy said, what is the problem we're trying to solve? And so very much in the inflammation space, if we think about dermatology and other indications, despite some reasonably new entrants such as the JAK inhibitors, I think we all recognize that having therapeutics that can be used chronically, long-term, that are safe, is still an unmet need, very much in dermatology and of course in other systemic inflammatory conditions. And that's still very much a focus for a lot of companies. Inforderm's play is in the BET bromodomain space, which is, I think, a relatively mature target class, but one that has struggled, I think, to get that ideal combination of efficacy and safety. And what the team has done from Dundee, and this has been a project that's been worked on for a few years inside the excellent Drug Discovery Centre up in Dundee, where, of course, quite a few spin-outs have been coming out of late. There's a team there of experienced, not just academics, but a team that's worked in that CRO space. They really know about delivery, and they've developed small molecules against BET bromodomains that have a clear difference versus the likes of GSK and one or two others working in this space. That's part of our secret sauce. I'm not going to go into that too much today, but we're small molecules. They have the right selectivity profile that will be efficacious, and from what we've seen in recent publications from some bigger players, have the right selectivity profile. So I think we're leading a new race in the bromodomain space, knowing that the selectivity profile isn't what people thought was ideal a few years ago, and we have that. So we're small, there's only 4 of us. We're whatever virtual means in COVID times, but we do have a small lab up at Dundee that's just reopened again, but we're very much in the outsourcing model, having just closed the seed round. We're delighted to be working with O2H, Meltwind as seed investors, along with Wren Capital. So that was closed this week, actually. So we're delighted to have done that. That sets us up for the next 12 or so months to deliver our program through to candidate ready. And with that, we'll be looking at Series A next year, alongside potentially some partnerships with some larger companies.
Tim, thanks. That's great. And I'm glad you mentioned Dundee. It may not be known to everyone, but Dundee has a fantastic drug discovery pedigree and heritage. And how do you feed off that? And where exactly did the discoveries sort of come from for this?
I think that the genesis, as you say, there's been an excellent, kinase group up there for years. And I remember Merck, we collaborated with Philip Cohen's group back in the day. But there's been a long-term strength in drug discovery and development. But I think the crucial difference here was that the team, and Andy Woodland and the others, they come from that CRO land, having gone into academia. They bring that hardcore experience of delivery of chemical matter that's developable into that academic setting. It's a very healthy fusion, coupled to, of course, the strength in tropical medicine and neglected diseases that Dundee has. But for this team, a very clear focus in inflammatory disease. And so all those hallmarks and strengths of Dundee, I think, have been a wonderful forming ground for this idea and this way of getting chemical matter with a differentiated profile.
I know you've said in the past, being first in an area is not always the best place to be. And you've mentioned already that, that bromodomain area is one that's been, trodden before. And so sort of why now? You said that there's a view of a change of profile. This often happens. People develop for a target and then discover it should go into a different area, or they discover that actually the ideal, what's often considered early single selective profile may not be the ideal thing, and profiles change. How did that come about, and how is that? I mean, I obviously can't go into too much detail, I imagine, in terms of, for intellectual property reasons, but, how did that change? How has the landscape changed?
Well, the group was working on this idea of a different selectivity profile within the bromodomain class. And then, sometimes fortune favors the brave, and a couple of publications have come out highlighting the issues with the unselective compounds in this class, where you can clearly get efficacy in a range of therapeutic areas, but the price is a very narrow therapeutic index. What we're seeing with our data is that with a different profile, you can get that efficacy you need, but seemingly without that narrow therapeutic index. And that will be very much core to our onward delivery, where we maintain that profile as you get compounds that we can develop. And I think the last thing to say there is that, and I mentioned it earlier, dermatology is what we've set out to do, and I think the clue is in the name of the company. But at the same time, if we can get bioavailable compounds, then generally anti-inflammatory approaches could be applied to other areas. So our challenge is
and you're going oral, topical, or both?
It's going to be oral, although we do have a separate program that's topical as well. That's got a different profile, and I think we may look at partnerships to take that forward while we focus on orals for derm, but also other areas.
Fab, Tim, thanks. So talking about therapeutic windows and selectivity and targeting, I'm delighted to introduce Miriam of Spirea. Miriam, do you want to just walk us through that problem and all the way through to where you currently are.
Yeah, thank you, Andy, and hello everyone. So yes, so at Sparia we are focusing on developing the next generation of cancer therapeutics. As Andy mentioned, they are called antibody drug conjugate. So these drugs are shown to have worked, been used for cancer patients on lymphoma, breast cancer, for instance. So there are now 9 approved ADCs, and they are really working very well for some patient groups. So the way they work is they are a combination of a small molecule and an antibody. So they have this antibody targeting cancer cell, which is providing the selectivity, and onto the antibody you have a drug payload, which is a cytotoxic attached, and this is done by a linker. So they are quite complex molecule, but very efficient in getting to the cancer cell and delivering the payload to the cancer cell. Now they've been struggling. So in terms of efficacy, they're quite good at doing their job, but they've been struggling with toxicity. So what's happening is those drugs have a constrained format. They have a format whereby you have a limited number of cytotoxic drug that can be attached onto the antibody. So usually that's what we call the drug to antibody ratio, or DAR. So you have usually a limitation of 2 to 4 cytotoxic molecules that can be attached. And this is limiting the way that they would work, because the payload, if you want to keep the activity of your ADC, which is coming from the payload, the payload must be very potent and therefore very toxic. And if there is premature release of your drug payload, or if the ADC binds to healthy tissues, this is causing some serious side effects. And this is limiting the therapeutic index of your ADCs. Also, so far, most of ADCs are limited to patients or populations having high expression level of the antigens. So meaning that even though they are really great molecule, powerful molecules, they have a lot of limitation and they cannot be applied to many cancer patients. So what we do at Sparia, we have a platform that we're going to apply to develop some drugs. And this platform allows to reformat the ADC and increase the number of drug payload, of cytotoxic payload that can be attached onto the antibody. So we can increase the drug to antibody ratio. So we are effectively developing high-DAR ADCs that are keeping stability. And this for 3 main benefits. The first benefit is the drug payload can be less toxic, less potent, therefore less toxic. And this is key to improve the safety profile of your ADC and the therapeutic index. Secondly, the payload mode of action can be matched to the target indication, so to have a better sensitivity for certain tumor. And lastly, high-DAR ADCs have shown to have a better efficacy for tumor expressing a lower expression of the target. So meaning that those ADC can treat more patients across more indications, so really more patient groups. So where we are in our journey, and so maybe I'll just highlight a few ADCs which are now showing this change in paradigm in the ADC field. So you have 2 ADC which have been approved recently. So you have an HER2 from AstraZeneca, Daiichi Sankyo, and Trodelvy from Immunomedics, which are known as, well, high-DAR ADC, and have shown to have a better therapeutic index and treating more patients. But they have a limitation in terms of the payload they can use. So what we do at Sparia, we can vary that payload. We can really look at different payload and really match the payload to the target indications. So where we are in our journey is that we have now achieved platform validation with in vitro and in vivo proof of concept studies. So we have applied our platform to model systems, so HER2 trastuzumab, and we use a payload MMA. So we shown we can have some great efficacy in vivo. And now we're going to use that platform validation to start a pipeline of in-house and partnered programs. So really now is, okay, we've shown this is working. How are we going to use it? So what are the next unmet needs? What are the unmet needs that our ADC can tackle? Because we have this opportunity to significantly increase the therapeutic index of those drugs.
Fabulous. Miriam, thank you. And I think this is really, so I think this is really exciting because because of that just the change in the way you think by increasing DAR allows you to go for less toxic payloads, which sort of has to be an opportunity. But then this concept of being able to go for low abundance targets on cells, that was a revelation to me, and I think that's key. I mean, but you have therefore so many potential routes to go down, and that's why I imagine you go down partnering and developing yourself.
Yes.
But that challenge of working out which payload, which target, which cancer, that's a big one.
Yeah, no, that's a big one, and that's exactly what we are up to right now. So which is great because thanks to the Cambridge ecosystem, or the people we've been talking to, we're getting the expertise to select the target indication and the payload that are going to be used for that target indication. So we believe that there is a scope to address a lot of unmet need, and as you said, it's just about selecting the one that we think will have the best, the highest impact. And one way to do that is indeed have our in-house programs. So have really think through about exactly what we're going to do, but also work with partners. So with pharma or biotech company, which have been developing ADCs, but some of them have failed because of narrow therapeutic index. So the way they have a fantastic target and they think that they want to apply in an ADC format, but they couldn't get that safety efficacy profile for their ADC. So that's how as well we want to apply our platform as well.
And just remind me, you talked about normal DAR being 2 to 4. Where can you get it to?
So right now we have an ADC which has a DAR 16, 16. So that's one ADC molecule we've been working with, but we have all range. So that's something which is very interesting as well with our platform, is we have high flexibility around varying the DAR. So I was mentioning the Daiichi Sankyo, AstraZeneca, and Trodelvy. So they have a DAR 8. They cannot. I mean, they can go down, but they cannot go up.
Yeah. So this gives you another, in a sense, at a molecular level, dosing play.
Exactly, because we can do everything from, DAR 4 to up to DAR 16, and we want to see how far we can go with that. It depends very much on the payload, and it's not just about the DAR number is not what it's what you're going to apply to. So what is very, very interesting is we're going now to screen different DAR and different payload and see what is going to be the efficacy and safety profile of the ADC to have the best therapeutic index.
But biology being the way it is, and it's always more complicated than everyone thinks, I'm sure you'll find something interesting there.
Yes, indeed. Yeah.
Okay, great, Miriam, thank you. And finally, I'm really pleased to talk to Vid. Vid, you're a quantum physicist, I understand, who's decided to sort of move into our sort of world of drug discovery. So I'm fascinated to hear about that and the problem you're trying to solve in the journey. Yeah, over to you.
Thanks. So yes, a little bit about Quano, and I guess my background and how I came to work on sort of in the pharma area. So basically, the aim of Quano is to, our aim is to build an asset-based biotech that's powered by quantum and AI technologies. And my background is in quantum physics, so I worked for many years in sort of many-body quantum physics and simulations. It was mostly theoretical work, but geared towards chemistry and material science. And there's a strong overlap between what I was working on and also quantum computing. So it's kind of what we are essentially looking to be at the forefront of the upcoming quantum revolution and apply this to pharma. So that's kind of the high level picture. The kind of very specific focus of what we're doing is, so we're focusing on enzyme inhibitors. And the connection between enzymes and quantum is basically that enzymes enzymes are quantum mechanical. It's kind of one of the topics in pharma, so to say, where quantum really matters, where quantum mechanical effects really play a big role, and where often sort of standard chemistry approaches and standard ways of thinking about chemistry that maybe medicinal chemistry used to no longer apply. So this is kind of again, like a relatively high level reason why we're focusing on enzymes. So what problems are we trying to solve by leveraging quantum technology specifically? Obviously with many targets, but I'm talking especially sort of specifically about enzymes, the big challenges in pharma, sort of, as everybody knows, are selectivity, resistance, trying to identify sort of molecules that they're actually potent enough to reach the target or to be useful as drugs. So our sort of again, sort of relatively high level thinking is towards optimizing for these three properties, for better resistance, better toxicity properties via selectivity, and better discover, kind of identifying very potent molecules that can then in the pharma pipeline be optimized, for all the other properties that are necessary to eventually get a drug to market. And this is where we believe that quantum can really make a big impact. So the specific way that we're doing this is by targeting transition states of enzymes. So transition states are in some sense the most quantum complex, the most quantum complex aspect of what happens in an enzyme catalyzed reaction. So the combination of AI and simulation, that's part of our platform, is key to delivering essentially drugs that target transition states. So I can say a little bit more about what exactly we mean very quickly. So basically, we are on the simulation side, we are simulating the enzyme. We're deploying these sort of very complex quantum mechanical simulations in order to identify the transition state, and then once we have it, the aim is to find a transition state inhibitor, which is a stable molecule that maximally well matches the quantum properties of this transient state, the transition state. The reason for doing this is that it's sort of from a theoretical point of view, it's the most. It is the most potent inhibitors are transition state mimics. So that kind of ticks the box of potency, but also equally, transition states are really sort of, they can be thought of as fingerprints of particular enzyme reactions. So they're very specific with respect to particular enzymes, even within a family. So in other words, having the capacity to accurately target transition states opens up room towards much sort of better selectivity properties. And a similar argument sort of actually can be used to demonstrate that targeting transition state is indeed also the best way to maintain for the drug to remain robust to genetic mutations. And so that's kind of the starting point. And so many of these points have been, so it's not, we haven't sort of come up with this idea. We're just the company that's enabling this in silico for the first time. These ideas have been known for quite a while, and some sort of these properties of transition states have been kind of appreciated for a while. And the challenge is overall sort of taking this tech and then marrying it up to a relevant and commercially interesting target roadmap. And the key ingredient in this, really, the key exciting ingredient in this is not only that we're able to sort of develop drugs that are better, that have better selectivity, better robustness, better tox properties, but also the platform opens up the way towards first-in-class drug design, in the sense that, unlike many AI-driven methods, this approach doesn't really require a lot of data to be available. All we require in principle is information about the enzyme, and so we essentially replace high data requirements with the capacity to actually calculate this transition state. And so we've kind of carefully developed a roadmap that marries up the tech development that's necessary together with with sort of a direction that's towards really, I mean, this term sometimes confuses people, so I hope it's kind of clear enough. It's kind of, I'd say, post-translational epigenetic targets like DUB proteins. So that's kind of what we're focusing on very specifically.
Okay, I was gonna ask you where you were going. DUB seems like an interesting space to go. Been really challenging the DUBs area to find great drugs. So this is, I think this is very attractive. As someone who did a PhD in enzyme chemistry long time ago, but I do like this. And tell me, what do you need? What data do you need? Yeah, I mean, you need how good a 3D structure of the protein do you need, and how much information do you need about it? How it interacts with both the substrate and the products to be able to define what the likely transition state folding state is in your simulation. I imagine that's sort of what you do.
Yes, so probably our CTO would be able to answer this question in much more detail. So we need the crystal structure. That's definitely true, and it very much is target dependent, I would say. So we've basically so far we have as far as our where we are in our journey, we have sort of, I'd say, a strong prototype. We validated this on an existing transition state drug. On an existing transition state drug program where there is a lot of data available that we can kind of reproduce in silico. This was done sort of all in wet lab experiments. So basically, in that case, we were actually amazed how well the in silico platform did with relatively limited information. And then since then, we've kind of worked also on targets that have that are more sort of quantum complex and where there are sort of issues with crystal structure, and certainly homology models can also be used. So we can kind of also target focus on targets where there isn't directly a crystal structure, but we do need some information. And there's obviously a relationship between there's somehow kind of an inverse relationship between the amount of information that's available and sort of the quality of the output ultimately. So that's kind of roughly the answer. But we do need sort of data about the targets, obviously. But
yeah, there's a question coming in, and I'm going to ask that one in a second. I'll just keep going, and please questions everyone if you can. Yeah, one of the things I think that's interesting. I mean, we've seen lots of AI drug discovery. Companies starting an initiative, and very often coming from physics and maths side of things. One of the things you've done, seems to me you've done very early on, is you've put together a team that's not just from that side of things, but also has proper drug hunters and medicinal chemists. And I've always thought that was needed, because having done, been inside the depths of companies doing small molecule drug discovery, the theory from the outside and the way it says in the books is never quite how it happens in practice. And there are elements of black art. So I'm sort of intrigued what you think you've learned from that interaction, and then I'll come to your question, Joe, which I think is a great question.
Yes, so this is indeed probably. I mean, it really I'd say that, one of our key USPs is that we have an interdisciplinary approach to this, because it's incredibly easy to fool yourself as a theoretical physicist and think that you can solve something easily, when actually there's a whole host of huge complexities associated with the problem. And indeed, so the only way that we've been able to actually deliver, start delivering results is because we have an interdisciplinary team that's true. That's necessary sort of at all levels of our tech pipeline. So from the molecules that are being generated by the AI to make sure that those are as drug-like as possible, that they're not, that we are kind of ticking as many of the drug discovery boxes as possible as early on, to sort of details about the interaction site and about what's going on, and so we can control that as easily as possible. So sort of it's basically the approach, the technology is very far from a black box. In other words, it requires really a lot of domain expertise, and this also then relates to our target roadmap. So it's not the case that we can, just because we have the idea and we have the generic setup that we can now target,
No.
you know, huge swathes of potential disease targets.
Well, I suppose it usually works like that. but I'll ask you, and I don't know, you might feel that actually you need your CSO there for that but Joe Rennie's asked what I think is a great question. Presumably small kinetic isotope effects are helpful to arrive at the saddle points for a particular substrate. Because kinetic isotope effects would give you much more data as well. I don't know whether that's available for many of these situations.
Yes, so indeed, that's exactly how the sort of the wet lab approaches in the past, specifically by a group by somebody called Vern Frum, who's based in the I think it's called the Albert Einstein Institute. It's in New York. So he spent his career actually, you know, working on transition state inhibitors using these methods experimentally. Obviously that data. So in reproducing his results, we actually didn't use the kinetic isotope data at all. We just used the crystal structure and nothing else. So from that point of view, it's not necessary. All, you know, as far as sort of looking at the whole drug development program, the more data you have, the better. There's absolutely no doubt. I think it's also true that these experiments are very expensive and they're only effective on a certain small subset of enzymes. And then even if you do have the transition state sort of identified using these methods, it doesn't really help you develop novel, the inhibitors for more complex quantum targets. In other words, the approaches that are out there have only been have mainly focused on what kind of, as a physicist, I would think of as relatively easy, quantum easy targets, so to say. And there are a whole host of very interesting and very potentially valuable targets where the quantum mechanics is more complex and where these methods, I think, wouldn't be as successful. But
so thanks. I hope, Joe, that's answered your question. And more questions from everyone. I'd just like to sort of move away from the specifics of the individual technologies. And I know you're all sort of, gearing up for financing events of one form and another. It would be great to just get a take from you all on what the financing environment is like at the moment and how you're finding it. Obviously saying how wonderful O2H are in all of that, of course, but I think we've come through the pandemic with life science being much more interesting to people. I think there are a whole load of new players coming in. I think people were sitting on their hands for a while, but I think we're seeing a flow of really quite big investments and things now. So it's good to get a sense on that. Tim, do you want to start?
Sure, no, happy to. And I think it's a really interesting point in time, exactly for the reasons you've just said, Andy. And I think specifically for Infoderm, on the other hand, it's benefited from what is, I think, an excellent framework and environment in the Scottish landscape for biotech companies, where there is plenty of supportive mechanisms through funding and mentoring and innovation encouragement. And that comes from Scottish enterprise and other aspects of the environment. So really that was crucial in getting Infoderm going. But then, of course, that can only get you so far on your journey. And then with the sort of seed environment that we've just been through successfully, O2H is, of course, wonderful, exactly as you've said, for the reasons they've supported us. And they bring in not just the investment, but of course, drug discovery experience as well, which is important. Then, of course, with our other seed investors, Meltwind and Wren coming in, that gives us the sufficient capital to get to the next value inflection, as long as we deliver properly and so on, and then to our next round.
It's sort of interesting that, because classically, Scotland had this great bunch of sort of home angel groups who were it tended not to be specialist. Archangel, Braveheart, etc., who then got backed up by Scottish enterprise sort of money and got people quite a long way. I'm sort of intrigued at the fact that you've gone for what I would describe as life science specialists and not them set from Scotland. So O2H, Meltwind, and Wren and others. Your reason behind that decision?
I think, and you can turn it around in a way. I guess it's a recognition by specialist life science seed investors that what we have is differentiated. But on the other hand, it's an early stage drug discovery program, which brings challenges that we all recognize as in small molecules, that it's important to understand the product profile that you're really going for, so you really stay focused on that. So I think having knowledgeable life science seed investors in will help us, especially with the commercial challenge. As I said, we could just remain dermatology focused. On the other hand, other anti-inflammatory areas could open up, including rheumatoid arthritis. So I think having those knowledgeable investors will help us stay the course and pick the right indication, which we do need to do as part of our next stage.
Miriam, do you want to say anything about funding environment?
Yeah, of course. Well, I guess for Spiraea, I mean, it's. I think I would say, I mean, UK was, I mean, is great as a way of getting some, you know, early stage ideas emerging, you know, from university. So Spiraea is a spin out from University of Cambridge, and you know, thanks to Innovate UK grant and some match funding as well, we got early on from IP Group, and then Cambridge Enterprise. I mean, we got a good start, so that was really great to get that support that early in Spiraea journey and in our ideas of getting some ADC drugs. And then you know, and then I'm part of this great ecosystem that Cambridge, so the Judge Business School, and then we got support from the Healthcare Accelerator, Starcodon, and then Jonathan Milner. So you know, we've been, you know, building, you know, our journey through that really amazing connections, and now we're getting support from O2H, which is, you know, fantastic. And I think we have those now, this great you know, group of investors and mentors who are around us and have this ambition and vision to get Spiraea to the next stage, and you know, and Sunil, and you know, is really, you know, helping us getting as well out there, and you know, sharing our vision and ambition. So I think it's key as well for us for the next stage, because we want now to expand. We want to be out there in this big world. So and reaching out to investors outside the UK. But I think I would say that for us being in Cambridge and having all those people supporting us been just perfect.
Good. And good luck.
Thank you.
Vic, you're in that interesting space where my guess is, you know, you sort of can interact with what I've called on many times the two tribes and the bit in between. And I'm sort of intrigued to your view as to the different investor ecosystems that look at what you're doing and the different aspects of business model, which must be fascinating and not least a little confusing sometimes about which way to go.
Yes, indeed. I mean, kind of from the perspective of companies sort of that are based on quantum technologies, we're doing something quite unusual compared to most. So we're focusing on an asset-based business rather than a platform-based business, and that effectively means that we're building a biotech, and therefore most investors will treat us as a biotech company, even, you know, sort of.
Well, it's sort of interesting from all of the, you know, if you look at the companies that actually have really grown from the AI and drug discovery space, many of them started saying platform, and most of them have become program orientated with time. Now that may just be a function of how far they feel they need to go up the value chain, but you know, I think that does tend to happen.
Yeah, I mean, I think I really kind of strongly believe that kind of focusing on a very, very specific problem is the best way to leverage quantum technologies, rather than having a broad platform that's kind of doesn't actually bring too many benefits across the board in the end. I think that's kind of what you observe with many of the businesses that have tried to go down that route first and then kind of started focusing later down the line. And I think that's why sort of, you know, O2H is kind of unique and sort of has this sort of special capacity to support businesses like ours, because we are, you know, at the moment, you know, not a fully fledged biotech company, so we need sort of the biotech support, and indeed sort of the plans are that you know, we're looking to close around with O2H for around for £700,000, and then the sort of the conversations with tech investors are along the lines of following sort of, you know, following the biotech lead. And sort of in that direction, we've had sort of support from a program called Creative Destruction Labs in Canada, which is also quite interesting. It's a quantum program, but they've got a lot of sort of industry-specific expertise and also links to investors. And in addition to that, we have some sort of limited success with Innovate, but are also applying for Innovate applications. In fact, one of them is with the Dundee Group as well. So there's lots of potential in this like you say, I forget what you called it, but the sort of space in the middle, right?
Yeah. Yes, the convergence space between the two tribes. And questions, please. Actually, there are a significant number of people in the participants list, the attendees list that I know, and I'm almost tempted to name some and ask them to come out with their questions, because I know some will. So I suggest you ask your questions, some of you who I know, before I point to you. But Rowan, thank you. And I was almost, you were going to be one of the first people I was going to point to on this. And you pointed out that early investors in US in silico companies were very often start with deep tech investors and then cross over to life science investors. And the exceptions were those that were started by top tier life science investors from the beginning. And my guess is, I mean, you're sort of in this space as well. You probably know this. You know this very well, and I think that's an interesting thing. I mean, I guess the other thing, and it sort of applies to all of you, and you've mentioned this mix of sort of platform versus products and partnerships versus own development, and there's always a sort of capital element to this, and I guess you're going or going through that, and it'd be good to maybe explore that. But perhaps I'd just like to ask something which those of you who've heard these sessions know that I'm always quite obsessed with, and that's intellectual property. And I think it's an interesting question here, because, you know, my guess is, and I don't know what your strategies are, but my guess is, Tim, you're in traditional small molecule drug discovery. It's fairly conventional what you do in the early stages. You need to work at getting strong substance of matter, and then you can add layers going on forward. And you know, I'll let you comment on that, but my guess is that's where it goes. My guess for Vid and Miriam, you know, you can go on big IP hunts on the platform. You can wait till you've got the products, and I'm sort of intrigued as to what you're prepared to say about where you go down that track. Miriam, are you happy to speak to start with before Vid?
Well, I'll share what we see. Our thinking is so in terms of where we are in our IP strategy. So we have a solid patent. So we have patent applications now on the platform, right? So that then can be applied to develop some specific products and drugs. So we have a broad patent application covering that. Now, in terms of our product, I think, yeah, the ideas would be to indeed have composition of matter on specific products, which would combine payloads, you know, our linker and an antibody. So we aim to have that kind of protection. And I think when it comes to working with partners, so I know that can be a very interesting discussion to have with your partners. I mean, that's where it's good to have a strong team around you, because we have you know, working with Adam Collier, who's been working in business development in many
who I noticed was on the line.
Right, and we have Jane Dancer and Gaynor Friars. I mean, they've been all, you know, having those discussions. I mean, from as well the biotech and the pharma point of view. So that's, I think we'll get some good partnership and, you know, finding the best way to move on in the IP situation. But yeah, I agree, that's an interesting topic.
And Vid, your asset focus, so I imagine the ultimate aim is to own the asset, as you say, the program.
Well, yeah, for our own programs, yeah, definitely we want to own. We want that's going to be our product. And in terms of getting the partners, I think that's with the partners that's a discussion to have with them. How we're going to do that. But yeah, no, that's why we want to have our in-house programs, because we really want to as well, you know, have clear control about what we're doing with our molecules and where do we want to push them in terms of next stage. Yeah.
And so Vid, have you protected it in a sense any of the platform?
Yes. So we have.
Sorry, Miriam, I was asking Vid, if that's all right. Vid.
Yeah, so we have actually five patent submissions for the platform, and the idea is kind of to ring fence the set of technologies for this particular application. I think it's very difficult sort of to patent machine learning network or something like this, because very quickly there's so much open source stuff, and it's basically impossible to protect in a meaningful way. So it's this kind of ring fencing approach, and I think what's emerging, kind of our views are kind of slowly maturing in some ways, and what's becoming clear is like what's valuable and what's less valuable, where we should kind of what we should really focus on as far as protection, and very often it feels like there's it's the sort of target specific data and target specific technologies that are the most valuable, because you can, for example, have an completely open source approach to machine learned force fields that you're using in some part of the pipeline, but it's how you then train the data, the quantum data for a particular target, that you can keep that proprietary. So
how novel are the underlying quantum mechanical models, or are those based on very standard quantum mechanics?
So
by the way, that's not my question, that's Rowan Gardner's question. Try not to claim the cleverness of that question.
So basically, one way to think about this is that sort of the complexity of the target proteins, the quantum complexity of the target proteins is governed really by mostly by the number of sort of metal atoms that are part of the active site, really, or that are kind of impacting the active site. So the more complex if you have, the more zincs you have, the more irons, if there are any irons present or something like this, the higher the complexity, and the more non-standard the methods become that you're using. So we've actually patented some of the really some of the technologies that are that are necessary to tackle really the very hard targets that link up to quantum computing. That's kind of future-proofing us a little bit. But for some of the easier targets, sort of standard methods are sufficient. And sort of the space in between, again, is quite interesting, in that that's where you can actually leverage machine learning to speed up the quantum calculations, and that's where the methods might be sort of widely available, but the data that you use to train for a particular target that you've kind of generated yourself, that can be valuable and that can be protected. So.
And Miriam, can I go back? You might not want to answer this, but your ability to be able to use payloads that other people can't, and your ability to target, let's say, less abundant cell surface receptors, you tempted to try and patent those, or the application in those, or are you going to refuse to answer my question?
Well, I'm not sure how detail I can give, but yeah, indeed, I mean, as you point out, there's scope and opportunity here to look at payload, which people said, oh well, that's not going to work as an ADC format, and then you say, well, now as we can increase the DAR, indeed we can do that. So yes, there is scope here to do this. Yeah.
And Tim, I assume yours, and I'm sorry, I did just assume where you were going, and this is absolutely classic small molecule drug discovery, new chemical entities, everything. How do you actually search? I mean, how good are you at actually knowing what's out there?
No, it's exactly, you answered the question in a way. It's classic small molecule composition of matter IP, and Infodem, because of the work of Andrew and Mark, the founders, they've got a healthy portfolio already. So already into that prosecution phase. But really, at face value, there isn't a platform here as such, but there is exactly to your point, how do you go about finding the gaps in the existing chemical space? And that's part of what the guys do really well. It's to look at what's gone before, and with a topical application route in mind, to know what works and what doesn't work from a chemistry perspective. That is in itself a platform, but then also applying that knowledge to design developable small molecules is, in some ways, as you said earlier, a black art. And part of that's gut feeling and hunch, but coupled to, you know, experience of what you learn works and doesn't work. So it's kind of, in some ways, an old-fashioned approach. And you know, we did that at Merck, like many other big pharma companies, and patent hopping and busting is sort of a very lucrative way of...
It's been an approach that has been, let's say, rebranded and renamed multiple times, but often ends up as the same. Sunil, you've appeared back. Does that mean you're shutting us off? Or can I continue with a couple of questions?
Please continue, Andy. I'm enjoying it.
Thank you. So David Ford, David's asked, and it's a vid question. Could you comment on the competitive landscape in such what he calls a nascent area? And I'd be intrigued, first of all, whether you agree it's a nascent area, and secondly, talk about the competitive landscape.
That's for me.
Yeah.
All right. Yes, so I think the competitive landscape is quite interesting in this area, because there's a lot of companies working towards quantum computing, and that's the challenge there is that sort of quantum computing itself is probably for most applications 5 years plus away for as far as any sort of practical applications. And therefore, many of these companies are building the platform, or of course, if they're building the hardware, that's a different business. But for those that are building a platform, they often kind of, I think, do a lot of consultancy business. So they're quite different to what we're doing. As far as sort of focusing on a specific asset, that approach is quite unique, I would say. Especially in pharma. So there are a few companies that are focusing on OLEDs or maybe on material science problems, but not yet directly in pharma. And then there are companies also that are, again, I guess this sort of space in between is becoming the topic a little bit. So this space in between where machine learning can be used to accelerate sort of quantum calculations, that's becoming quite interesting. And there are companies that are building sort of mostly platform capabilities. And again, sort of the asset focus is not so much present there yet, but I think it probably will be soon. So that's kind of.
Did you have a look, or can you make a comment on the DeepMind claiming the protein folding problem last week?
Yeah, that's super interesting. It could be actually like a really useful technology to support what we're doing. According to Dave, my co-founder and CTO, so this is obviously everybody's very exciting, and it's a great advance. But as far as sort of the accuracy that you need to actually model the active site, it's not there yet.
No, it's not. And Shruti, I think Vid has answered your question in various answers about the AI tool used in the simulation studies. If he hasn't, then I'm sure, Sunil, can you put Shruti's question to Vid offline and get an answer? I guess in the last couple of minutes, I don't think, are there any other questions? Oh yes, there is. Joe's asked a question. Miriam, just a curiosity, how are the yields of the ADC impacted by increasing DAR? Just thinking about this from the manufacturing viewpoint, if this is a relevant question. I think it's a very relevant question. Miriam.
Yeah, no, that's a very good question. So so that doesn't impact, I mean, the yield. So I'm not sure which state we're talking about, but in terms of making quantities of ADC that you can test, that there's no bigger, the challenge is not increased in terms of manufacturing your ADC. What we had to overcome, which we did, it's about homogeneity. So when we do our DAR 16 is because we use a linker where we can attach multiple copies of a drug payload, and we optimize the manufacturing approach to get a monodisperse. So you have one linker payload copy, and then you attach that onto your antibody the same way as you would do it for I mean, existing linker payload. So there's no challenge in terms of doing this with our linker. So you use the same approach, but you just use a linker which has more copies of the drug payload. So that's something we've been manufacturing in quantity enough to do our in vivo studies, and that's all fine.
Great, thanks. So Sunil, I think we've slightly run over time, and we should let everyone get back to their emails and 4 o'clock Zoom sessions. My guess is that if anyone wants to speak to Tim or Vid and Miriam, they can do it through you.
Yeah, no, absolutely. We'll facilitate any introductions that
Great. And is there
anyone on the
anything you
team.
want to answer or say at the end, Sunil?
Yeah, look, I just really enjoyed the session. I think you facilitated that brilliantly, as I would have expected. It was very nice in a way to have the speakers come on board and have a very contrasting different, they're all working on different modalities, different technologies, but all in the biotech ecosystem space. And it genuinely is, I think all 3 of these companies could really go places. So we're very happy to be supporting this group of companies. And, I think the world's their oyster, really. It's a huge tailwind right now in biotech. I think the Zoom platform and COVID, in a way, bizarrely has opened us up to the world in a way that we've never had that access for a more global biotech ecosystem. I don't know what you feel, Andy, around that. But,
I think that's absolutely right. I tell you, it's got harder chairing board meetings, but that's a different thing. But
at least you can bring on the right expertise from wherever it is around the world.
Absolutely. It's been wonderful for actually bringing on and talking to people across the globe as if we were, close in a room and not having to go through that faff. And I got my invite today through for JP Morgan main conference, which of course is virtual this year. And I thought, not doing the streets of San Francisco in January. In some ways I miss it, as I know it's a bit of but in other ways, it's going to be wonderful.
Yeah, it's a love-hate relationship with that conference. We all love going to San Francisco in January and escaping the cold weather, but it is a zoo, as we all know, and having to, able to pick and choose what you see in the comfort of your own chair and home has this massive advantages. So it would be interesting to see how that continues next year as we come out of this.
So Sunil, I'd like to thank you for organising these. I think you've done a great job. I don't know if there's another one before Christmas, but beyond that, I'd really like to thank Tim and Miriam and Vid for subjecting themselves to the sort of knockabout of these sessions, and it's great to see you all. And thank you all on the for staying on the and listening. I think it's been great fun. And have a great weekend, everyone.
Thank you very much. Merry Christmas to everybody, if we don't have another one, which is quite likely. So thank you very, very much.
Thanks, Andy. Thanks, everyone.
Thanks, everyone.